In this video, we will be talking about your Francisella. What are the descriptions for Francisella? So it is faintly staining. It's a tiny gram-negative coccobacilli. As you saw in the picture on the last slide, they are very small. So it is oxidase and urease negative. And it is catalase positive. Again, oxidase and urease negative. And it is catalase positive. So it is non-motile, it is non-spore forming, and is a strict aerobe. You have four different species for your Francisella. You have your Francisella tularensis, Francisella novatunensis, Francisella novoside, and your Francisella philomiraja. So in this table, it's easier to memorize. So your Francisella species, it is named after the American microbiologist named Edward Francis. It is he who first described tularemia. Okay? So your next is also known as your type A or your Francisella tularensis subspecies tularensis. It pertains to the Tulare County, California, where the disease was first described. Next, we have your Francisella tularensis subspecies Holarctica. It is also known as type B. Holos meaning whole and Arctos meaning the northern regions. So it is a reference to the distribution in the Arctic or the northern regions. And then next we have your Francisella tularensis subspecies Media asiatica or your type C. So media meaning in the middle, Asiatica so in the Asian side of the world. So it pertains to the Middle Asia. Next, we have your Francisella tularensis subspecies novicida. Novus meaning new and sida meaning to cut. So a literal translation would be new killer. And lastly, we have your Francisella philomiraja. Philos meaning loving, miraja meaning mirage. And what it means is that it is loving of mirages or mirages. It is a reference to the presence in water. The Francisella tularensis is under the biosafety level 2. All subspecies require cysteine, and your Francisella tularensis has three subspecies. Again, this is your type A, type B, and your type C. So your subspecies tularensis type A, it includes three subclades, your A1A, A1B, and your A2. It is also subdivided into type A West, which is predominant in the arid regions of the Rocky Mountains of the Sierra Nevada mountain. Next, we have your type A East, which is predominant in central southeast states of the USA. It is most commonly associated with exposure to lagomorphs or your rabbits and hares and felines as well. So type A East is more commonly associated with disseminated disease and has a high mortality rate compared to the type A West. Next subspecies, we have your subspecies Holarctica or your type B. It includes 10 different subclades naman for this one. That is predominant in the upper Mississippi River and in areas with high rainfall such as in the Pacific Northwest. So it is most commonly associated with your exposure from rodents and felines. So mga daga and mga pusa. We now move on to the disease that it causes. It is tularemia. It is a zoonotic disease that can be contracted either through ingestion, inhalation, and contact with infected tissue or arthropod bite. So, an arthropod bite, is, or it exhibits a hard itch. And what you need here is that you need your Ixodes, Dermacentor, and Ambyloma. So these are the drugs. The drug of choice for your tularemia is your gentamicin. Tularemia has 6 different types of infection. First, you have your ulceroglandular, which is the most common. It forms ulcer in the site of inoculation and your lymphadenopathy. So it is rarely fatal. Next, we have your glandular. So it involves only the lymphadenopathy and it is also rarely fatal. Next, we have your oculoglandular. It is a specialized form of the disease and results from direct contamination of the eye. So it involves conjunctivitis and your lymphadenopathy. Next, we have your oropharyngeal. So it involves the ulceration of the oropharynx. Next, we have systemic or your typhoidal tularemia. These involve an acute illness with septicemia and has a mortality rate of 30 to 60%. These have no ulcerations and or no lymphadenopathy. Again, this is for your systemic or your typhoidal tularemia. Then next we have your pneumotic tularemia. It is acquired by inhalation of infectious aerosols or by the dissemination from the bloodstream or simply from pneumonia. So it is a serious form of tularemia. Next, we have your Legionella. Legionella was first recorded during an American Legion convention held in Philadelphia. So in the year 1976. So it comprises of 52 species and 70 serogroups. They are ubiquitous, they are an obligate aerobe, they are poor staining, they are motile, and they are gram-negative fastigious bacilli that require a medium supplemented with irons and L-cysteine and also buffered to 6.9 for the optimal growth. So this organism utilizes protein rather than carbohydrates for your energy source. Your Legionella species in clinical specimens do not stain with common reagents, but it can be seen in tissue stained with diethyl silver stain. Okay. Out of the 52 species, only 20 species are pathogenic to humans. First, we have our Legionella pneumophilia. So it has 16 serogroups. It is oxidase, catalase, and hypurate hydrolysis, and gelatinase, and lastly, beta-lactamase positive. So it is considered as a facultative intracellular parasite. Legionella pneumophilia exists in two well-defined, morphologically distinct forms in HeLa cells. So a highly differentiated cyst-like form that is your highly infectious. So it is metabolically dormant and is resistant to antibiotics and detergent-mediated lysis. Another is a replicative intracellular that is ultrastructurally similar to agar-grown bacteria. Its virulence factor consists or includes your heat shock protein 60, your outer membrane protein, your macrophage infectivity potentiator, your type 4 pili, flagella, and the genes encoding for the type 2 secretion systems, and your DOT type 4 secretion system. Pontiac fever is an influenza-like illness which is self-limiting and is febrile. It was first described in 1968 at Pontiac, Michigan. Okay, so the symptoms are fever, chills, myalgia, malaise, and headache. But there are no clinical signs of pneumonia. So this is caused by your Legionella pneumophila serogroup 1 and 6, and your Legionella phylae. Another disease cause is your Legionnaire's disease. So this is having pneumonia as your prominent manifestation. So it is the one of the top four causative agents in community-acquired pneumonia. So Legionella produces different symptoms from your strep pneumoniae. For your Legionnaire's disease, your serogroup 1 accounts for most of the cases. So the dissemination via the circulatory system can lead to extrapulmonary disease with or without pneumonia. The disease manifests in three major patterns. First, as a sporadic case, and then an epidemic outbreak, and your nosocomial clusters. So, for nosocomial, as you all know, this occurs in compromised populations only. Your laboratory diagnosis, you use buffered charcoal yeast extract with PAV or PAC, your polymyxin N, anisomycin, and or either vancomycin or cefamantol. So colonies examined with autofluorescence. Your colonies are blue to white or yellow, green, or red. So that is the autofluorescence of your colonies. This is a summary or a table for your colonial autofluorescence.